Our model provides a much-needed experimental system to investigate what's happening during the window of implantation in humans. This will revolutionize what we know about this crucial time period in pregnancy, providing lots of exciting avenues for researchers to follow up, and ultimately to help us understand more about the biology of how we develop and of common pregnancy disorders, and perhaps also improve the success of IVF. I'm Peter Ruggan. I'm a senior group leader at the Babraham Institute near Cambridge in the UK, where I lead a research team within the epigenetics program. The research themes within my group involve studying how epigenetic information is first established in human development and how this information controls how embryos grow and develop. This is important to study because epigenetic information helps regulate which genes are switched on or switched off in the right cell types at the right moment in development. In my group, we use a combination of human stem cells, embryo-like models, and embryos that have been donated to our research in order to understand the molecular pathways that control the initiation of epigenetic processes. Our new discovery provides the first approach in the laboratory for understanding how early-stage human embryos implant into the endometrium to establish a healthy pregnancy. The model that we've created contains all the major cell types that are found in endometrial tissue. And what we've done is we've managed to layer these different cell types in a way that recapitulates how the tissue normally looks. From analysing the morphology of the cells within the model and looking at their behaviours in terms of gene expression, we can see that we'd managed to engineer a system that closely modelled the complex cellular architecture of the human womb lining. So as part of this, we were really excited to see that our system also produced essential factors that are normally needed to nourish the embryo in the first few weeks of pregnancy. Previous models haven't been able to achieve this, and so this really represented a breakthrough for us, and it told us that we were moving along the right lines. After checking and validating our system, it was time to see whether it was capable of supporting the implantation of a human embryo. To do this, we used surplus human embryos that were donated to our research from individuals that have undergone IVF treatment. And we were really happy to see that a high number, about 3/4 or so, of the embryos successfully implanted and were followed in our study until 14 days after fertilization. We've already been able to observe some never-seen-before aspects of human embryo development in its relationship with the tissues mimicking the womb lining. Although the embryo is microscopic at this early stage of development, using specialized microscopes in the laboratory, we were able to visualize the shape and the structure of embryos in our model system. Excitingly, this matched what we know about embryo development in the womb at this stage, and this was great because it told us that the embryos were developing faithfully over this short period. An important aspect that we wanted to understand in our study is what are the signals whereby the embryo first communicates with the endometrium when they meet. Until now, it's just not been possible to decode these signals at this early developmental stage, but with our new research, we were able to listen in, so to speak, and to understand what these signals are. We believe that these may be the same signals that tell the embryo and the endometrium to keep going and to establish a pregnancy. And perhaps these signals might be missing in some pregnancies leading to embryo loss. There's a lot of research ahead of us. So far, our research has only used endometrial tissues from healthy donors. But in the model that we've created, it could be adapted in the future to use endometrial tissue donated by individuals who suffer from conditions such as recurrent pregnancy loss. This will allow us to understand in these individuals whether there might be differences in the way that embryos and endometrial cells might signal to one another.